Okay, hi guys, welcome to Biology Online with Henny van Vieren. Okay, hi year 12s. Welcome to this next lesson in the life processes at a cellular level section. Today we have to look at enzymes and specifically how they function, what they are, and so forth. Now, you might remember from last year and potentially many years before that enzymes are basically biological catalysts. So what is a catalyst? A catalyst is something that enhances or enables a specific reaction to take place without physically taking part in the reaction. In other words, the enzyme can be used again and again, or the catalyst can be used again and again for a specific chemical or biochemical reaction. Now, the first thing that you need to know is that all enzymes are proteins. Okay, they are all proteins. All proteins are not enzymes, but all enzymes are proteins. Now, proteins, we get two types of proteins, filamentous and globular. Okay, so the one is like the globfish. Okay, glob. It is a ball of stuff that is a specific shape. And the shape of enzymes is extremely important. So, and then filamentous. If you have a long filament, like a piece of fishing line is a filament, that is a single strand. Okay, now we also get long proteins called filamentous proteins, and that's usually used in structural things and in muscle fibers and so forth. Now, oh yeah, there you go. Filament is usually structural, like collagen and gluten. Okay, globular are highly folded. The shape is integral to the function. Now remember earlier we touched on the lock and key mechanism, where, and we'll touch on that again today. But it's the shape of that. If the shape is not right, then the substrate or the thing that it has to help react won't fit, and consequently the reaction won't take place. Now, globular, these usually include things like hormones and enzymes and so forth. And as I said earlier, enzymes are biological catalysts. Catalysts enable or speed up reaction by lowering the activation energy to take place. Okay, so bringing them together, making sure that their orientation is correct and that there is enough energy for the reaction to take place. Okay, so here we have a very high level example of, or a representation of an enzyme. Now, if you think that this blue thing is made out of a single strand that has been folded and folded and folded and folded and folded and folded and folded until it created this shape with a very specific active site. And this active site is complementary to the substrate that it's going to help to either have it create, or in this case it's going to break them apart. Okay, so think about it as the lock and the key. So this is the lock and there's the key, and as long as that can go in, the reaction will take place. Okay, but that's why the shape is so extremely important. Okay, so again, our ribosome, this, we're back at the endoplasmic reticulum, and this is the rough endoplasmic reticulum because it's got the ribosomes. The ribosome makes a polypeptide. Now, if the polypeptide is never folded, or folded very infrequently or just a few times, then we'll call it a filamentous protein. Okay, in this case, the polypeptide. From here, it goes off to the Golgi apparatus. And in the Golgi apparatus, or the Golgi body, that's where the folding and shaping takes place. So here's an example of a very long polypeptide, and the polypeptide needs to be turned into something like this on this side. Now, you can see that this is the catalytic site, or the active site, and only very specific things will be able to fit in there. And they call it a binding pocket, but you can look and see kind of the idea that the people had when they created this representation of something that is more similar to that. Okay, and that's why the Golgi apparatus, when they fold it and package the protein, is exceptionally important. So there are two types of enzyme reactions. The one is anabolic, okay, and Anna banana, anabolic, she likes to build things, okay, it needs energy. and then we have catabolic. The cat likes to rip apart your couch in the living room, and that is, it releases energy. So catabolism is breaking down, anabolism is putting together. Those are the two reactions that enzymes will work on. Okay, now the active site, or the location where an enzyme binds to a substrate. As I said earlier, it is very, very specific. They will only bind to specific things because of their unique active site. Okay, so in this case, your rectangle will not be able to fit into there. It has to be the triangle. Now, there's two existing theories, and this frequently comes up in the exams where they want you to describe the two existing theories and why enzymes are specific. So, for instance, if you have an enzyme that codes for, I don't know, eye color, that's going to code for the protein that creates the color of the eye, and it suddenly starts to produce that enzyme in the color in your hand, and you suddenly have blue eyes, or you understand what I mean? So it has to be specific where they work. That's probably not the greatest example, by the way, but never mind. So initially, scientists believed that the active site was like a genuine lock and a genuine key. Well, not genuine, but very, very stringent. In other words, they have to be exactly complementary. The fit has to be like a lock and key. It has to be really precise. Now, that is the old way of thinking. The current way of thinking is that we have more of a induced fit theory. Induced means to make it fit theory. So it is very close at the beginning, and then when the new one comes along, it'll change its shape to allow for the new fit. Okay? So enzymes are flexible. The binding of the substrate molecule to the enzyme molecule modify the shape of the active site so that it becomes complementary to the substrate. So here's an inactive enzyme. Okay, it currently looks like that. Now the substrate comes along, and you can see the substrate is a hexagon. It's not a half circle or not a full circle. So how can this enzyme get that to react? It induces its shape from this. Okay, and you can see this one is a star, so it will change its shape to accommodate. Now when it's activated, it changes its shape, and bang and bang, and you have a new molecule that is formed. Okay, so that is the induced fit model, where they don't have to be an exact match, but they will the enzyme will change its shape to accommodate the substrate. Here's another visual of that. So with the lock and key, they have to be a precise match. With the induced fit, you can see they are much more malleable, and they can consequently make the reaction happen. Okay, so when we think about an enzyme process, and we're going to do this later on in the year, you can almost think of it, every one of these factory workers, they are an enzyme. So if this guy over here doesn't do his job properly, down the line somebody else won't be able to do that properly. Or if these guys who put the engines into the cars over here, if they don't do it, you will not have cars with engines in them so that the next guy can work on it. Or if this robot back there breaks, then the next robot won't be able to do its job, because it will, then you have a buildup of product, etc. So think about an enzymatic pathway as a rolling production line. Okay. Yeah, and this was actually invented by Henry Ford when he was making the first mass-produced automobile, which was called the Model T. Not the first car, but the first mass-produced. Okay, so every one of these enzymes are like little workers that make things happen, but you don't see them become part of the car, so I hope that helps. Now, because an enzyme is a protein, the same thing that will affect your proteins will affect your enzymes. So these, a few of the things, so these can be things like pH or solvents, okay, and salts. These are all chemicals that can influence it. Other things can be like toxins, or sometimes toxins can be salts or solvents. and so forth. So when you have an optimum temperature, pH range, then your enzymes will function optimally. But as you go further away from the optimum range, the lower their rate of reaction will be, because they won't be happy. And what happens when you put, for instance, some of you might have seen this, but if you take, you saw the video of the naked egg, I hope, but if you take a small quail egg and you put it in vinegar for a very long time, the low pH of the vinegar will actually physically denature or cook, if you want to call it that, the egg. Now I know that the Maori have a and also Samoan and Tongans have a lot of fish dishes where they use lemon juice to cook the fish. But what they're basically doing is they're denaturing the enzymes, not the enzymes, but the proteins in there with a low pH. Okay, so it's all about the optimum pH, or then your optimum salt concentration. That's why a saltwater fish and a freshwater fish will struggle if they change environments. So when it is too high or too low, it denatures. Now what does denaturing mean? With your molecule, and I'm wondering if there's a picture, that's not a great picture, but anyways, when your molecule is this long filament that has been folded and folded, there's little hydrogen bonds between the folds of the filament. So think about a string, and we take the string and we're wrapping it and 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 wrapping it, and we want to make an active site that is somewhere there. I don't know. Yeah. Okay. Now, wherever this kind of the lines come close to each other, there's going to be a hydrogen bond. Okay. And these hydrogen bonds are the ones that break when the pH is not right, or when the temperature is not right, or when you make pavlova, for instance, when you take your egg beater and you beat it and beat it and beat it and beat it and beat it, you're also changing the actual physical structure. So because of that, the shape of the active site changes more and more and more and more, and the further it changes, the less active your enzyme. And when it denatures, this is not reversible. When it denatures, it's gone. Okay. Anyway, so look here at the optimum range of pH. So enzyme activity. Pepsin is something that comes out of your stomach. It works in your stomach. So look where it's most active. Right here at a pH of 2. So you can obviously tell that pepsin, stomach enzyme, or enzyme that helps to break down food in the stomach, wants a very acidic environment. Something like amylase, which we find in our saliva that breaks down starch, you can see that it loves a very neutral pH. And then arginase, you will find that in your small intestines, is more towards where we have a more basic environment. Okay, so different enzymes will work at different pHs, also different temperatures. I spoke to you about extremophiles. So extremophile organisms that love to live at very high heat, their enzymes would work at very high heat, and so forth. And this is where the next one come on, factors that affect enzyme, like temperature. Now, if you take an organism and you cool it down a bit, it's actually going to slow down its rate. Okay, you're slowing down the kinetic energy of your physical enzyme and also the substrate. Now, this does not necessarily change the shape. You're not freezing them, you're just cooling them down. It doesn't necessarily change the shape of the actual enzyme or the substrate, but it slows down and takes away the energy, so that when you think about particle theory, now you have fewer collisions per unit of time, so the rate of reaction will become lower. And because there's fewer collisions with enough energy and fewer collisions with the right orientation. So that means less successful collisions. Alright, now when that heats back up again to the optimum range, we will see the enzyme activity pick back up. So that is reversible. When it's too cold, it is reversible, and this is very important. Okay, so around 37 degrees Celsius, the body's temperature for humans, this is where we do our best. Now, when you go to above this, This is where we get denaturing happen. So again, we talked earlier about taking a whisk and you're whisking egg whites for a pavlova, but now you're not whisking it, but you're cooking it, and you're also physically changing it, because egg white before you cook it is translucent, or no, transparent, or one of those kind of see-through, and it becomes white. And the reason why it becomes white is because those proteins physically change. You're physically changing their shape. So the same will happen with an enzyme, and when we change their shape physically, because we're breaking those hydrogen bonds, our binding site will change shape, and consequently we won't work anymore, and that is a definite problem. Okay, you cannot return that. I've got a good friend who, unfortunately, when he was little, he got a very, very high fever, and because of the high fever, his optic nerve got damaged, and he's blind today because of that high fever. So humans, our body runs at about 37. That's where we want it. If ever you have a temperature of above 40, you need to get into a lukewarm bath. You want your bath to be at around 37 degrees Celsius, not a cold bath, because a cold bath will do more damage than good. It will drive all the hot blood just into the middle of your body and nothing on your skin. But if you have a 37 degrees Celsius bath, you open up the vessels in your blood, in your skin, and a lot of blood can then come into contact or very close proximity to the water, and you can lose a lot of heat. So remember that, please, whenever you see somebody with a very high temperature, put them in a lukewarm bath. Definitely not a hot one, and also not a cold one, a lukewarm bath. Okay, keep that in mind. It works very, very well. I've done it many times with my kids. Okay, so that's the end of enzymes. I hope that helps. And yeah, we will also have a look at some stage on the SciPad, but at the moment I've assigned you a task and also a few assessments. Now the task itself I think you need to do first, and it's all about factors affecting enzymes such as pH, salt, temperature, and so forth. So please have a look at that, and yeah, good luck out there, guys. Keep safe. Thanks a lot. Bye.